Oh, I need to. I'm going to share over here. Hang on. What is that? Where are you coming from for traffic?
From Raceland.
From race.
About 25.
Make that. Okay. Yes. Yeah. And this is, we were talking about, yeah, good. That's where we were. Yeah, I know there's a bunch of people come today.
Oh, so it was?
Yeah. Oh, yeah.
I don't know.
There's a bunch of people coming today. No, I think I put it like 8 hours. Yeah, no, I know I've got a bunch today. Sometimes they do. I don't know. I just like I just wait for my thing to tell me somebody's coming, somebody's coming. But all right, so we're working on the question from last time, right? Everybody's working on that. And then you picked up a sheet. You need to keep that sheet. We're going to work on it throughout the chapter. So today and next time. And it is also on Moodle. Let me pull it up on Moodle. So if you're on Moodle, it should be at the end of chapter 16 stuff. I go 13, 14, 15, 16. So chapter 16 questions. Okay. So it'll be a little while. We'll have a few minutes to finish working on that. Y'all are pretty close though, right? Sort of, kind of, maybe. No, clicker is not on. I need to turn clicker on. Thank you. Anybody else need another handout? Everybody got theirs?
I need another one. Thank you.
Yeah. So we're working on what's the motivation for this? That's the one we're working on right now. I know I gave you another one, but we're working on what's the motivation for this. And I'm starting iClicker. I know I'm behind. Okay, so make sure you're checked in on iClicker. Make sure you're checked in on iClicker. So we're doing right now, what's the motivation for this? And then didn't get it, we're gonna use this handout later today. And you have one of these in Okay, I'll start. New page. Guess we can separate it, right? Let's see. Is the other handout? Yes, the other handout is on Moodle. So one of them, I don't think I can, is right there. What's the motivation for this? And then the other one is right underneath chapter 16 questions. So that's the chapter 16 questions. And then it's so weird. And then what we're working on right now is what's the motivation for this? Okay, good. Good. All right, so fatty acid synthesis and oxidation, right? Okay. All right. Yeah, do share. What have y'all done? Oxidation or synthesis?
We've done a lot.
You've done both?
It's a good question.
I should have brought my coffee this morning. I thought about stopping, but I was like, I don't have time.
There's always time. You can always be late.
Y'all, and what would, but if I walk in with just coffee for me, that would not be nice.
I'd be okay.
If I were late, y'all would be like, peace out. We're gone.
10 minutes away.
Take your time, really.
Honestly, I would have gone and picked up the coffee.
Yeah, you're supposed to talk to another group. You're supposed to compare.
Oh, no. No, man.
I want to hide.
Yeah, we did oxidation. We thought everyone else was taking something.
Oh, right, sure. Good to know I'm not the only one struggling this morning.
Yeah. Classic pick up a phone.
I don't know why.
Do it. Wait, I have no idea why. If I put the curve, it's probably better if it doesn't draw.
But then it's sunnier.
Nothing.
I think around spring. You don't like the warm weather.
Oh, no.
Oh, no.
It's
Oh,
not.
yeah, no, sometimes.
Oh, wait.
I don't have hot girls in my hot girl winter. I'll try to ride in the fall.
Like the general public moves in just a
That's
little
not.
bit to pick up the surface. I was literally just doing this.
All right, I'm gonna give y'all like 5 more minutes. So y'all need to get to it. Y'all crack me up. Chapter. Book. They might have a Khan Academy, something like that on there, Sam. I'm not sure. Have you tried the animations and stuff from the chapter?
You look and you see it just fine. Oh, no, not the little thing.
Yeah. Well, hopefully this will help today. At least you'll get the answers. Getting close, right? Because I hear some off topic.
Everything becomes bright.
2 minutes. 2-minute warning. All right, looks like everybody's on a different topic. So y'all ready? All right, so let's talk oxidation first. Talk oxidation, because what are the 3 things that we were asked to do for both oxidation and synthesis? 3 different things. We're looking for reactions that what? That either use or generate high energy metabolites, right? So we can classify these really as exergonic and endergonic, truthfully, right? So let's kind of separate it into exergonic, endergonic, and then you were asked one other thing in the handout.
Let's see high energy molecules.
Well, that's endergonic and exergonic, right? If you're going to generate one or if you're going to consume one. But then you were also asked about regulation, right? Which stages are regulated for each of the pathways? And then kind of overall, why, how do you have these 2 competing pathways, right? And how do they not have futile cycling? Because we've talked about that for like glycolysis and gluconeogenesis, right? Okay, so what do you get for something that is exergonic for oxidation? Exergonic is hydrolysis of ATP, right? If you have ATP and you're going to hydrolyze it into AMP and what? PPI. All right, we're going to get energy from that. Okay, now what? Now what about PPI? Pyrophosphate. Correct. So PPI goes to 2PI. That's a nice negative delta G value, right? Okay, what else? It got quiet all of a sudden. What else? Talk about oxidation. So what are some other oxidation things that happen? What are you oxidizing? Well, so you're in the oxidation that is breakdown of fats, right? So exactly what is oxidized? Yes, but that's actually endergonic. Energy of endergonic. In endergonic, we're going from, we have a reduction of NAD plus to, well, NAD plus and FAD, right? We have both of them because we're generating both. What else do we generate? Acetyl-Coa. But these are our reduced substances, right? Yes? With me so far? I feel like we all need a really big coffee. Okay, so what about, let's go back to exergonic. What else is oxidized? You're oxidizing some carbons, right? Right? So we're oxidizing the carbons. So these were carbon-hydrogen bonds, and they're oxidized to what? Carbon-carbon double bonds, right? And then what else is exergonic in terms of, think about disorder, right? If you're starting out with one really long fatty acid, what do you end up with? Smaller ones, right? Exactly. So what's happening to disorder as we do that? So we go from one large molecule to many small, which is an increase in disorder. Y'all get anything else for exergonic things going on? Okay, how about endergonic? I only have one other thing at endergonic. Anybody? It's the water. Yeah, incorporation of water. So this is our hydration step, right? Okay, so before we go on and do regulation, let's go and do synthesis, the same thing for synthesis. Trying to keep this separate. Okay, so exergonic. What do we do? It's similar. When we synthesize fats, what do we have to hydrolyze? Generation of fats requires what? Well, yeah, but what high energy molecule, right? We're going to consume, just like in over here, we're going to consume what? ATP. ATP. We have to invest ATP, right? So we have ATP hydrolysis. Okay. So now, what else? When we're first making fats, what do we do to it? We prime it. Once we prime it, what do we have? Yeah, what's it called? What's our substrate called? It's something CoA. The fatty Acetyl-Coa. So when we do that, in our next step, what happens? Yeah, that's when it can go into the mitochondria. But that's degradation. No, it's not transport, non-transport. What happens when we have malonyl CoA? When we generate malonyl CoA? We're going to release something. What are we releasing? I thought I heard somebody say it. It feels like a Monday today. We release carbon dioxide. We lose that carbon. Go back and look at the pathway. We lose the carbon. So CO2 release. Ooh. From malonyl CoA. Yeah. Maybe. What else? What else do we oxidize? Because we oxidize in oxidation, but we also have things that we oxidize during synthesis. What substances get oxidized in synthesis? The opposite of the things that are reduced over here, right? So we're going to oxidize not NADH, but NADPH. Right? This is another one of the ways that we're able to separate oxidation and synthesis. One of them is going to use NAD plus and NADH. The other is going to use NADPH. Okay. All right. What else? Maybe else get exergonic. I sure had a lot of talking. What happens at a dehydration step? What do? We release something. What do we release? Water. Okay, so that's what I have for exergonic. What do you have for endergonic? So think about things that are reduction. Okay, a condensation reaction. Yeah, so condensation. Why a condensation reaction? Because in a condensation reaction, what happens? We have many molecules turning into one larger. Very good. Go back up here. Reduction of, what are we reducing? Yes, yeah, you could put that. NADP plus. What else? In the fat that you're synthesizing. Yeah. The carbons, right? The partially oxidized carbons. PART, partially oxidized carbons, right? Y'all have some of this? What you were writing? No? Well, and you could talk about the individual steps, but what's important to kind of keep track of is that in both cases, you have reactions that are exergonic and endergonic, right? It's not just like synthesis is requiring energy and breakdown is giving off energy, period. You actually have both in both reactions, right? It's not always an expenditure in synthesis and always a release in degradation. I'll see where we're going with this, possibly. Yeah. Okay. So let's talk about regulation. Tell me about regulation and oxidation. What's my regulation step? The carnitine. The carnitine is actually our regulation step. So regulation at carnitine transport. N-I-T-I-N-E, carnitine. And we call it a shuttle system, right? Carnitine shuttle. So if we don't shuttle it into the mitochondria, is it going to be degraded? No. Right? So when do we not want it to be degraded? if you have a high energy charge, you are synthesizing fats, right? So we don't want to degrade them. So if we are synthesizing fats, what is going to inhibit carnitine shuttle, the carnitine shuttle system? Correct. BIT pitted by malonyl. Whoa. CoA. Okay. So how about our regulation in synthesis? Mhm. Of what? Acetyl-Coa Carboxylase. So how do you make either synthesis? I can move it up. Oh, and Sam, I'm sorry. I haven't been monitoring the chat. So how do we make sure that both of these reactions can happen at the same time? You're always going to couple things that require energy with things that produce energy, right? So just like in oxidation, we are coupling the energy requirement of breaking those bonds with what? Think about your first step. We're going to prime our fat to be degraded. What investment does that require? An investment of ATP. Well, but that's investing energy. So how do we make that favorable? We couple it to pyrophosphate degradation, right? So you're always going to have that coupling of things that require energy and things that consume energy to make either synthesis or degradation happen. Doesn't matter. You always have coupling happening. Yeah. And then you also have enzymes that are regulated differentially, right? Like phosphorylation, dephosphorylation, right? We have compartmentalization. So maybe you want to add this, right? So you could go compartmentalization, CO. So we have, what did we talk about? We talked about coupling, favorable, unfavorable, reactions. What else did we say? Oh yeah, so differential regulation. Okay. And then are you going to use exactly the same enzymes to go one direction as you go the other direction? No. So that would be enzyme specificity. I think those are the big four. How you can have competing pathways for no futile cycling. So to prevent futile cycling. Anything we forgot? Anybody want to add something? No? Okay. So we ready to go back? Y'all got it? Y'all want me to leave it up for a second? We got it? Oh, and I didn't tell y'all jokes today. So here's a couple that Charles did not understand whatsoever. What do you call a group of rabbits hopping backwards? A receding hairline. It's like, don't worry, bud, you will eventually. It was terrible. What do you get from a miniature cow? Condensed milk. Yeah, he didn't get that one either. He didn't get that one. All right, y'all ready? Get back on track. All right, so do you remember last time we were talking, we said there were kind of two big problems that can occur in fatty acid degradation. What were the two big problems? What were the two big problems? If you have having a trans bond, right? So what happens if you have a trans bond? What do you have to go through? An auxiliary pathway, right? Okay, so what's our other problem? Goes along with the other enzymes. Cis trans bonds. Odd numbers. Odd numbers. So what happens if we have odd numbers? So here's an example of a fat down here that has an odd number of carbons. Well, you're going to get some number of acetyl-CoA units out of that fat, right? No matter what, you're going to get some acetyl-CoAs. So what happens is that you get acetyl-CoA and you get this propanyl-CoA. So propanyl-CoA is a 3-carbon unit. A 3-carbon unit is not going to do us any good. So what are we going to do? We want to convert it to succinyl-CoA because succinyl-CoA can enter the citrate cycle. How many carbons is in succinyl-CoA? Anybody know? Anybody remember? From citrate? 4. It is a 4-carbon. It's a 4-carbon. So how do we just go from a 3-carbon to a 4-carbon chain? We have to add something to it. What are we going to add to it? A carbon. Where do we get carbon from? Like we just can't incorporate carbon dioxide, right? Like plants can. So we have bicarbonate. We have bicarbonate, and it's going to require an investment of ATP. So now succinyl-CoA can enter the citrate cycle. That's awesome. Now we have enzymes that can work on it. So within the citrate cycle, we're going to convert succinyl-CoA to malate. Malate is actually going to be shipped out into the cytosol, and in the cytosol, malate can be converted into pyruvate. Now that we have pyruvate, pyruvate can get shipped, guess what? Back into the mitochondria. And now we can actually use it in metabolism, in breakdown, right? It can go through the normal glycolysis to citrate pathway, right? And we can use it to generate energy. So you have to understand that, yes, most of the fats in our bodies are going to go through the normal beta oxidation in order to get through all of them. So most of them are even numbers, but they're not all even. And we can handle fats that are an uneven number. Okay, so if you, for example, had 23 carbons, that's what our fat is over here. You're going to eventually get 11 acetyl-CoAs. Eventually. Because that one propanyl-CoA will get converted to succinate, eventually back to pyruvate, back to acetyl-CoA. So you're going to get 10 out this way, and then 1 here. Right? So you get 11. So here's 10, and here's 1 for a total of 11. Trans or cis, either way, Sam.
The one at the very bottom?
Yeah, it's 23. This one is C23. All right, so now we know we can handle fats. So exactly what happens in the body if we limit our carb intake? We're going to go through what process? Ketogenesis. So there are kind of a few ways that you can do this. You can either go through, it's basically the starvation pathway, right? That's a low carb diet. Or if you have diabetes, it's the same, you're going to have the same sort of process. So what are we going to increase if we go low carb for whatever reason? We're going to increase breakdown of fats, which is what do we call that now? Beta oxidation. So in the liver cells specifically, we have excess acetyl-CoA, right? That's what beta oxidation got us. Lots of acetyl-CoA. So that acetyl-CoA is not going to go through the citrate cycle. Our citrate cycle is going to slow down. Why would our citrate cycle slow down? No oxaloacetate. And we have a depletion, DEPLE, depletion of oxaloacetate. So where's oxaloacetate going? If you haven't had carbs in a while, which organ of your body is throwing a fit right now? Your brain. Your brain wants glucose. And preferentially, your body will make glucose for your brain as long as it humanly possibly can. So how is your body going to make glucose if you're not consuming it? So the oxaloacetate is being shuttled out due to gluconeogenesis. So that y'all can see. So you could put brain under that, right? That's the really important thing. Okay, so if we don't have oxaloacetate, then what are we going to do? We're going to start creating what are called ketone bodies. And ketone bodies are made in the liver, and then we export them to basically almost all the cells of the body. The brain can actually use ketone bodies, but it doesn't like to. So first it's going to use glucose if it can, and then eventually it'll shift over to using ketone bodies, but it's kind of a last ditch effort. Okay, so let's look this is kind of just what we've been talking about, right? So here, start down here at the bottom. In our citrate cycle, what we normally have, right, we're going through it, but we deplete the intake of glucose, right? So we're going to shift our oxaloacetate, take it out, turn it into pyruvate, and be able to use it in gluconeogenesis. We've made glucose, we export glucose to the brain's happy, right? But now we have to worry about all the other cells of the body. So where are we going to get that? Fats from adipocytes are going to be shipped out. Liver is going to pick them up, going to get transported into the mitochondria via what? Carnitine, right? That's our carnitine shuttle. Now that we have fats inside of the mitochondria, we're going to go through beta oxidation. What product do we make? Acetyl-CoA. Acetyl-CoA can then go through ketogenesis to make ketone bodies. The two primary ketone bodies are acetoacetate and D-beta hydroxybutyrate. These two can be shipped out and go wherever they're needed. Okay, so what we're going to talk about is basically this step right here between the acetyl-CoA and the ketone bodies. Okay. All right. So here's our formation of ketone bodies. So what you're going to start out with in this process is acetyl-CoA. So we are going to combine 2 molecules of acetyl-CoA to make acetyl-CoA, right? So what gets kicked out? If we have 2 molecules of acetyl-CoA, each of them has a CoA unit. And if we combine them, we only need how many? 1. So we lose one of the CoAs. All right. Now we're going to take a third acetyl-CoA and we're going to incorporate a third acetyl group into our chain. So again, we kick out what? CoA. All right. So we're not quite there yet. This is HMG-CoA. I bet you've heard HMG-CoA before. Like maybe not knowing what it's for, but this is a substrate that's used for cholesterol synthesis. And we're going to talk about cholesterol in the last section. Okay. So now we have 3 acetyl units, but it's not in a substrate that we can use. So then we're going to go through a lyase step where we actually cleave off an acetyl-CoA unit, right? So we invested 3 so far. We're going to cleave out 1 acetyl-CoA unit. So now we're going to end up with acetoacetate. And the acetoacetate actually ends up having only 2 acetyl units within it, right? Okay, so now acetoacetate is one of these substrates that can be used for energy as a ketone body, can be exported directly. But there are 2 things that could happen. That acetoacetate can go through a reaction where we make the beta hydroxybutyrate. This is another substrate that can be used for energy. But at some rate, this happens enzyme dependent here and spontaneously. It shows it with an enzyme, but it will happen spontaneously. Well, you'll lose a carbon and you're going to make acetone. So anybody smell acetone? What's it smell like? Smells like acetone, right? So if you have a patient who comes in and they're displaying symptoms of ketogenesis, which we'll talk about, one of those things is that they have a very fruity smelling breath, and that's from the acetone. So it's one of those diagnostic features that you can use in order to know what's going on in the person's body. Okay, so now we have 2 substrates that can be exported out and be used for energy in cases of very low glucose in the body. All right, so now what happens to those ketone bodies? Well, those ketone bodies can actually undergo degradation if they can enter the citrate cycle, right? So how do they enter the citrate cycle? Well, they're really based on 2 units of what? Acetyl-CoA, right? So if we can take these 2 and we can turn them back into acetyl-CoA, then we can use them for energy, right? So that's exactly what we're going to do. We're going to convert them back to the acetoacetate, then to acetoacyl-CoA, and then split them apart. And now we again have 2 acetyl-CoAs, right? So now we have acetyl-CoA. Well, acetyl-CoA can enter citrate, and we can go and make energy. So this is the like the keto diet, right? Now I've heard about, everybody's heard about the keto diet. Okay, yeah. So one of the things that you have to kind of understand is that this pathway in the body was really designed for starvation conditions. It really wasn't designed for us to be on a keto diet for a really long time. So you have to be very careful about that. Yes. Well, yeah, and we're gonna, when we get to amino acids, like we're gonna get there. We're gonna absolutely get there. No, not in this chapter. Not really. But we will when we talk about amino acids. Yeah. So how is it that this does not happen? How is this not futile cycling? Why does this not degradation not happen in the liver? Well, why wouldn't it get broken down before it was exported? relatively stable. It's relatively stable. It'll go through your bloodstream, get to other organs. Where did we export it from? No. We exported fats from adipose. That's fine. Fat from adipose. Then where did the fats go? Where? Of what? Of the liver. So to liver. Liver is what's going to make ketone bodies. Then they export to, we'll say, muscle, because that's the primary consumer of ketone bodies. So it's important to kind of recognize what cells you're in, what organ you're in, right? So if the liver makes the ketone bodies, why is it that the liver won't metabolize the ketone bodies? You're actually missing the enzyme that will convert it back from the ketone bodies into acetyl-CoA. So this enzyme is missing from liver. So liver can't use ketone bodies for energy needs, right? Can't do it. It can convert back and forth between the two ketone bodies, but it can't metabolize the ketone bodies for energy.
What is the purpose of converting it to beta hydroxybutyrate, whatever, just doesn't need it?
It's more of a resonance thing, that it's a relatively easy process to happen. And so some cell types will use one ketone body over the other, but either one's fine.
So it wouldn't have to do with the production of NADH?
You do make NADH, but you don't necessarily need it. It depends on the cellular conditions.
Right, so if NADH was low, you would want to convert it.
Yes, if NADH is low. It resonates. Hmm?
It uses an enzyme, so like, what is the purpose of the enzyme in the first place? Because the enzyme requires NADH, and then it produces NADH.
The enzyme requires NAD plus.
Oh, no, to convert from acetoacetate to beta hydroxybutyrate.
Correct. If you want to go backwards, yes.
So you don't technically need that enzyme at all.
You could do without it. You could. Okay, so let's talk about the liver again, right? You're going through low carb, right? Your liver can't make an energy source from ketone bodies. So what is your liver going to use for ketone bodies? For ketone bodies, for energy? What is it can break down its glycogen stores, right? Okay, what if your glycogen stores are gone? What's the liver going to do? What can it do for the brain? Gluconeogenesis. That's me. Sorry. Okay, so it can do gluconeogenesis, and then it's going to also export ketone bodies for the rest of the body. But why? So now, why is ketone body export not necessarily always a good thing? So our problem is that whenever we make these ketone bodies, if you get too much ketone bodies, what's actually going to end up happening is you're going to have a very acidic environment. What do we call that? Ketoacidosis. You've made so many ketones and the body can't consume them all and the body can't use them all, and you go through ketoacidosis. What's the hallmark of ketoacidosis? A low blood pH. So you'll have nausea, you'll have vomiting, you'll have stomach pain, and you'll have fruity breath. If it gets really bad and you are hypoglycemic, if it gets really bad, you'll go into a coma, right? You won't be able to be woken or anything. Okay, so look at your little section question, right? Describe the benefits of ketogenesis, and then why is it dangerous to give insulin to an emergency room patient with severe ketoacidosis before first checking the patient's blood glucose levels? I want to answer that right now because it's like fresh on your mind. So why is it beneficial? You're starving, right? Your body's starving. So you're providing an energy source for the body. Absolutely. That's great. Okay. So look at the second part of the question. Like we just talked about the physiological benefits. So now, why is it dangerous to give insulin to an emergency room patient with severe ketoacidosis before first checking a patient's blood glucose levels? If you have someone who's diabetic, right? Tell me about that person. So they would end up with high blood sugar, right? Okay, so if you have someone who's diabetic, they have really high blood sugar, so what do you give them? You give them insulin, right? And it'll decrease their blood sugar. They'll take in the glucose and they'll be okay, right? But what if you have somebody who has really low, they're hypoglycemic, not hyperglycemic, right? If they're diabetic, they're hyperglycemic, but they're not, and they have sugar, they're just not taking it in. And so they go through ketoacidosis. But what if you have somebody who has all the symptoms of ketoacidosis, right? And normally you think, oh, it's a diabetic patient, I'm just going to give them insulin. What happens if their blood glucose is really low? They're hypoglycemic. You'll tank their blood glucose even lower, right? So you're going to do more harm than good. So what do you need to do if you think someone is in ketoacidosis? What's the very first thing you want to check?
Blood sugar.
Blood sugar. Then you know what's the right treatment for that patient. Yeah, you don't know. You don't know. You need to know that before you decide what to give the patient, right?
Like even like a diabetic person could have high blood sugar but still be going through ketogenesis because they're not using that blood sugar.
So what if you have someone like that who is really hypoglycemic? What are you going to give them? Well, I wouldn't even give them oral glucose. I would give them
Straight to the IV.
an IV. I would give them an IV
Just that way it gets.
or an injection, something quick,
Gets into the blood.
as fast as possible. As fast as possible. Yep. Okay. All right. So pretty good. Yeah, it could kill them. Yes. So pretty good. Yeah. Good. Okay. So now we're going to talk about synthesis. I can't believe it took us this long to get to section 2, but that's okay. It's all good, right? It's all good. Time to be had. Yeah. All right. So in the last chapter, right, we talked about different ways to get fats, right? We said you can ingest them. And what else did we say? How do you get fats? You can ingest them, dietary. And what else? What? No. How are we going to build up fats in the body? We could eat them, right? And then we store them. But we also said your body is always preparing for winter, right? Winter is coming. So if I eat carbs, what's my body going to do? It's going to convert them into fats, right? So in the liver, you'll convert to fats, right? Anytime in your liver where you have high glucose levels, so you have high glucose, and you've exceeded the energy requirements of the cell, you're going to start prepping for winter and you're going to start making fats, right? So our carbon substrates from synthesis of fats and triacylglycerides are from carbs, right? Yes, y'all remember that? I'm a little scared right now. It's okay. All right, so let's talk about what happens. If we have glucose, glucose comes in, what do we convert it to eventually? Like if I want to make fats, what do I have to eventually convert my glucose to? I'm going to go through glycolysis, and I'm going to eventually make acetyl-CoA. I'm going to move the acetyl-CoA to where? Like what organelle am I in? Glycolysis occurs in the cytosol, yes, but then I have to ship it to the mitochondria. So I'm eventually going to get to the mitochondria, right? So once I'm in the mitochondria, what is my acetyl-CoA going to go through? Citrate. Okay, so that's a nice little reminder, right? Before we look at a really big diagram. Let's go look at a really big diagram. All right. So here's our, I love that they did bagels. It's like, oh yeah, can handle that, right? All right, so we've eaten our carbohydrate source, right? We break down our carbs into simple sugars. We import them as glucose. This is what we already said we did. We go through glycolysis, convert to pyruvate, pyruvate into the mitochondria, pyruvate to acetyl-CoA. So we ended up with acetyl-CoA in the mitochondria, right? All right, so what happens to the acetyl-CoA? Well, if we have a high energy charge, right? This is, I'm having a bagel, but I don't really need a bagel, right? My energy charge is still high. So I'm going to convert the acetyl-CoA to citrate. But here's where I'm stopped. Why am I stopped at citrate? I have high ATP, right? And I have lots of oxaloacetate, right? So I'm going to inhibit the rest of the citrate cycle. So then what happens to citrate? It gets shuttled out. Citrate is going to go into the cytosol. Our citrate is converted into oxaloacetate, right? Okay. Now, when we convert from citrate to oxaloacetate, what do we lose? We lose an acetyl-CoA unit. How many carbons in an acetyl-CoA? 2 carbon units, right? Okay, so now we can convert our acetyl-CoA to malonyl-CoA. If we add a carbon, it's not really carbon dioxide, right? It's really bicarbonate, but that's cool. Malonyl-CoA is a C3, right? Okay, so now that we have malonyl-CoA, we can go through our fatty acid synthase enzymes and we can make fatty acids, right? But what's the other part that we need in order to make triacylglycerol? We need our glycerol backbone, right? Because that's what it is, a glycerol backbone and 3 fatty acids. Well, once we went through glycolysis, we can divert some of the fructose 1,6-bis phosphate to making dihydroxyacetone phosphate, which can be converted into glycerol. So we combine our glycerol with our fatty acids and we can make triacylglycerol. So the other thing that's cool is that once you have that citrate and you pull out your acetyl-CoA, the other product is oxaloacetate, which gets converted to malate. Malate goes back into the mitochondria and we have oxaloacetate, which can then be used to convert our acetyl-CoA to citrate. So everything's used, everything is reused over and over and over again, right? But this is our link for glycolysis, for citrate, for fatty acid synthesis. This links everything together. Isn't that kind of nice in one, right? So this is why when we eat carbs, we make fats, right? It's not just if you have a high fat diet. Having a high fat diet, yes, is going to lead to accumulation of fats, but so is having a high carb diet, right? Difference is how you move the fat to the adipocytes, right? Is it dietary and solubilized and then we package it, right? Or do we actually synthesize it in the liver, right? Good. Okay, so I want to show you, oh yeah, this is the other thing I was supposed to do, but that's okay. Check out the animation. The animation is really nice. So this is a really great comparison for fatty acid degradation and fatty acid synthesis. When we compare the two, right? Do you see that you have the same basic 4 steps that occur when we're degrading fats, and we have 4 steps that occur when we go through synthesis? We're doing kind of the opposite, but this illustrates really our big differences, right? We compartmentalize them. Where's degradation? Mitochondria. Where's synthesis? Cytosol, right? So we're compartmentalizing. What substrates do we use for oxidation reduction in degradation? We use NAD plus, and FAD. What do we use out here? NADPH, right? So we have different substrates, so that's nice. You have different enzymes in degradation than you do in synthesis, right? So we have that enzyme specificity. And we have different rate limiting steps. Our rate limiting step for fatty acid degradation is what? Transport into the mitochondria via the carnitine transport shuttle. And what about for fatty acid synthesis? Is our creation of malonyl-CoA, right? So if we come back up, our rate limiting step is where? It's creation of malonyl-CoA. But we know that malonyl-CoA is in the cytosol is going to inhibit what? Carnitine. So if we are synthesizing fatty acids de novo, right, we're going to inhibit import of fats into the mitochondria to be degraded. Woohoo! Right? We're not going to do fetal cycling. Yes.
Do we have a slide on malonyl-CoA, or did we just mention about it?
I think we just talked about it.
There's one of the slides that does.
Yeah, somewhere it does. Somewhere. But I don't think there's like a slide on malonyl-CoA. I don't think so. Okay. All right. Good. All right, so how do we synthesize fats? So we have one huge multifunctional enzyme that is used to make all of these fats. So we have what's called an acyl carrier protein that is going to anchor the hydrocarbons. So in the book, you're going to see ACP. That's the abbreviation for acyl carrier protein. So all of these acyl carrier protein, that's what the ACP stands for. So we know that the rate limiting step here for the generation of malonyl-CoA is through what enzyme? Acetyl-Coa Carboxylase, right? Okay, so we have a 4-step reaction that occurs in this multifunctional enzyme. The first one is ketoacyl ACP synthase. It's going to be a condensation reaction. And when you see these abbreviations in the textbook, they're going to abbreviate them KS. So where does the KS come from? KS, right? Then they're going to abbreviate KR, KD, right? And I think it's ER. I think that's the abbreviation on that one. So you'll see the book uses all kinds of abbreviations, and I think it's really kind of hard to keep track of. So what you're going to see is this 4-step reaction that's going to happen over and over again so that we can build a long chain fatty acid, right? So it's repeated in cycles, adding 2 carbons at a time. So repeated in cycles, 2 carbons at a time. Okay. All right, so where are we synthesizing? What subcellular location are we at? We're in the cytosol. Okay. What are we going to use as our reductant? From the last slide, what we've talked about the differences. NADPH. Okay. What is our first step? What's our first product? What are we going to make? Malonyl-CoA. All right. And that's our rate limiting step. So if we look, right, here's where we're going to start. We're starting with just 1 molecule of acetyl-CoA. So that acetyl-CoA gets hooked to this multifunctional enzyme. So do you see how they say acetyl-KS? What is the KS? So this is one part of the multifunctional enzyme. It's this one really big enzyme, and it's just like what part of the enzyme is the acetyl hooked to. And in this case, it's the KS. So kind of keep track of that when you're looking at the enzyme. All right. So we have acetyl-KS. So we're going to convert acetyl-KS into malonyl ACP. What is the ACP? There's so many abbreviations, it's terrible. Acyl carrier protein. Why are we going to call it an acyl carrier protein? What did we just do? What do we have in here? We have acyl groups, right? What's an acyl group? Carbon bound to a carbonyl carbon, right? That's our acyl group. Okay. So we undergo a condensation reaction and we're going to lose the carbon on the very end. So we have malonyl-CoA, which is our 3-carbon molecule. We're going to add it to acetyl-CoA, which is 2 carbons, but we're going to lose one of the carbons from malonyl ACP. So that 1 carbon is lost as CO2. So now what are you left with? 2 carbons from acetyl-CoA, and then 2 carbons that came from malonyl-CoA. So we still have a total of 4 carbons. 4, right? 3 carbons, 2 carbons, lose 1 carbon, and we end up with 4 carbons. Yes? So that's a condensation reaction. So think about we're going through creation of something really big. So one of the reactions that we said in synthesis is condensation. Condensation is going to give us that small molecules into one big molecule, right? Okay, so now we have to reduce our substrate. Go up a little bit. So now we're associating in each of these steps with a different portion of our multi-unit enzyme. So when we did our condensation reaction, what part of the enzyme completed that condensation? KS. So, and remember, you can always go up here and you can say, okay, what is KS? It's the keto-acyl ACP synthase. So now we're going to go through the reductase. Okay, so our KR is the reductase, our reduction. Then we go through a dehydration reaction where we're going to lose water. And in our dehydration, it's DH. It's not KD, it's DH. It's DH. Go back and fix that. So the keto-acyl ACP dehydratase that they do as a DH instead of KH. Okay, so that's this one, the dehydratase. So now we're going to take this one and we're going to go through a reduction step. We go through a reduction step and we're going through the enol reductase. That's the one they abbreviate ER. We have stayed in the same enzyme through all 4 of these reactions. It's just one huge multi-unit enzyme, and then we give the pieces a different name. Okay, so in order to do this, now we can go through this cycle over and over again and make each carbon, well, each fatty acid chain longer by 2 carbons, because that's in essence what we're doing. So if you look at the beginning, right, you're starting out with something that is how many carbons long? 2 carbons. At the end, how many carbons do you have? You have 4 carbons, but you have a substrate where you can now add more carbons to that substrate. You can go to 6 carbons, then 8 carbons, and so on and so on. And that's why most fats are, guess what? Even numbered, because this process occurs by adding 2 carbon units. Okay. So all the carbons are derived either directly or indirectly from acetyl-CoA. Right? Good. All right. So if we look at our acetyl-CoA carboxylase enzyme, right? So what are we doing for our acetyl-CoA carboxylase enzyme? What are we trying to make? We're trying to make malonyl-CoA because we need malonyl-CoA right up here in order to start adding acyl groups to it, right? That's our starting material, is our malonyl-CoA. So our malonyl-CoA kind of looks like a heart, yes. It's nice to see something pretty, right? So in order to do this, we've talked about this. We've talked about the acetyl-CoA carboxylase reaction one time before. What's going to happen first is that we're going to use up an ATP. Remember that? So we have an attack of a bicarbonate molecule on the phosphate, right? And so we're going to pick up that phosphate group on the bicarbonate, right? So we have carboxyphosphate. So this reaction requires biotin. So this is the biotinylated enzyme. So biotin is one of those required vitamins, right, that you need to take in your diet. So then you undergo a binding of acetyl-CoA, right? So we transfer that carbon-containing unit from the phosphate group to the acetyl-CoA. And so now we have created a 2 carbon unit. How many carbon unit, y'all? A 3 carbon unit of malonyl-CoA, right? So our malonyl-CoA is 3 carbons. Our acetyl-CoA started out as how many carbons? 2 carbons, and then we're adding, it was really over here, this bicarbonate that's right here, that is how many carbons? 1 carbon. So that's why our very first one, we need malonyl-CoA to be 3 carbons. Okay. So the way that this works is you have this kind of transfer of the enzyme, and the enzyme kind of works like a clamshell in order to transfer everything, but you don't have to worry too much about the enzyme structure itself. I'm not testing you on that. Okay. So now we need to talk about our acyl carrier protein, right? What function does our acyl carrier protein have? What is that? A lot? Yeah. but what's our what's our goal of our acyl carrier protein? To transfer acyl groups from what to what? What's that? So we do need a hydrocarbon anchor. We need something that will hold on to our fatty acid as it's being built, right? What's that? So it anchors our fatty acid as we continue to grow it. So I think before we start talking too much in this in detail, I think we'll end it here because it's 10:19, and we'll talk about our acyl carrier protein structure when we come back. So we'll finish 2 and finish 3, start and finish 3 because 3 isn't bad. 3 is just cholesterol. Sounds good? All right. You're going home now. This is the end of your day?
I got like